very comprehensive introduction. I will, I guess, go a bit more into my background, and also at the end of the Q&A session, if you want to ask me any questions about my journey, starting PhD, doing research, things like that, doing a startup, I'm very happy to answer your questions on things that are more personal. So today I'm going to talk about synthetic biology in the age of artificial intelligence and all the amazing ways that AI is advancing the field and contributing to our understanding of biology. So I'm not too sure everyone's background here, so I'm going to try and keep a lot of the topics hopefully pretty easy to understand, and I'll do my best to explain things. So as was said, I did my undergraduate degree at the University of British Columbia in Vancouver. I did research in very diverse areas, which was really good because it allowed me to learn all sorts of different things, and also helped me figure out exactly what I was interested in. So I did things ranging from working on gene editing technologies all the way to machine learning or even electron microscopy of plant biology. So it was quite the range, and I'm really lucky that I got to experience so many different things in research. And then after that, I moved to California to start my PhD at Stanford, and I finished my Master's of Science here, and at the same time, I've been doing my PhD in bioengineering. And I decided to pursue research in machine learning here because it's such a valuable field right now, as I'm sure is very obvious to everyone. And hopefully after today's talk, you'll understand a bit more why. So I'm going to go over the just high-level concepts about how deep learning works, and then from there talk about language models, what they are, and how we use them for biology. And then I will go more into the ways that language models and machine learning is being used specifically for synthetic biology. So we've seen very, very rapidly in the past couple years that AI, especially generative AI, has been advancing extremely quickly. This is an example of an image of a steampunk animal generated by Midjourney in 2022. And then in 2024, just 2 years later, how much more detailed and intricate the generations are. And this takes a very, very good model in order to generate an image that is this stunning and creative in different ways. And we're seeing this not just in image generation, but in all fields, how AI is advancing very rapidly. So the huge explosion in AI has been attributed to deep learning specifically. So deep learning is a subset of AI. If you consider artificial intelligence as just the idea of creating machines that can do things that require some level of intelligence, machine learning within that is more about the algorithms that humans can program in order for machines to do that. And then deep learning is specifically a part of machine learning where we use a model called artificial neural networks. So what are neural networks? They're based on the brain, actually. That's why we call them neural networks. It came from insight as to how neurons in our brains specifically communicate and respond to the environment. And this is this structure of a typical neuron where you have the cell body and it receives some input signals from other neurons. It fires along its axon, and then it outputs those signals through the terminals to other neurons around it. And so computer scientists thought we could build something similar with computers, except make some artificial neuron that receives inputs from other neurons that get multiplied by what we call a weight, which together gets added into the receiving neuron and then gets activated by some function and then outputted to other neurons around it. So it's very similar to the biological neuron, except it's just the computational version. And I want you to remember these specific terms like weights and bias, which get added to the sum of the inputs going into the neuron. These will be important later for understanding. So if you scale this up to a full artificial neural network, it's supposed to resemble what a chunk of the brain might look like, where you have this very dense architecture of neurons communicating with one another. Um, and you get all of these interesting circuits that can perform different intellectual tasks. And so the artificial neural network at the beginning has some input layer that where the inputs pass through all of these different neurons and then go through what we call hidden layers, and then finally an output layer where you get your prediction or generation. And this remarkably worked extremely well and is what allowed machine learning to advance so rapidly all of a sudden. So when training a neural network, the process involves gradually tuning these weights and biases that I just mentioned until your error on your training and test data that you're training the model with becomes minimal. So you do this iterative process where you pass training samples through the model. So for example, images with some labels on them or something, they pass through the neural network and the output is some label that the model predicts. And then an error is calculated as to how close that label was. And then that error is used backward through the neural network to update those weights and biases to create all of these very nonlinear specific functions all the way through the neural network that eventually learn to capture the distribution of the training data. So again, that every neuron built in this artificial neural network is composed of these units where inputs are passed, the weights on each input is multiplied and added together and then activated through a function and output to the next neuron. And it's the weights and biases here that are being updated through this training process iteratively to get better and better. And the way they're updated is through this algorithm called gradient descent, which is absolutely foundational to training machine learning models. And it's this idea that with gradient descent, you're calculating partial derivatives backwards through the network that allow you to find the direction during training where you're moving to lower and lower loss or error on your training data. Hopefully this isn't too confusing, but this is the general idea behind how pretty much all neural networks are trained today, and it's the foundation of deep learning. And once you've trained a model that has really good ability to predict or classify, then you'll take that model and you'll just freeze it in the state that it's in. So it has frozen weights. And now this is the model that you'll use for all of your other tasks. So if you have a different image that you want to classify through it. So this process is called inference when you're starting to make prediction on your inputs with your final model. And here, if the image is like a human face, then the model has now, instead of predicting it as a dog, it predicts it as a human face accurately. The variation of neural networks that we use for natural language are called language models. And the main idea behind these is that you want to predict the next word given the previous words or the previous part of the sentence. So if your input is once upon a, for us, it's very obvious that the next word would be time. This is a very common expression used in many stories. But for a model, you need to train it in order to learn that that's the next most likely word. And so the goal of language models is to learn a probability distribution around language with enough diverse training examples that allow it to figure out all of these different functions through the neural network that can guide you from one input to the most likely output. So the general expression for this is the probability of word n given all of the prior words behind that word that you're wanting to predict. So if we wanted to sense every word individually, let's say in the English alphabet or English language, there would just be way too many lines or connections in this dense neural network. It would very, very quickly become extremely inefficient and require too much computational power. So a big insight in building neural networks was to create what we call embeddings, which is a compressed representation of your inputs through the model at different layers. So this can make the model architecture more streamlined and efficient, where it learns to take inputs and shrink the dimensions of those inputs down into a smaller space that is much more computationally easy to handle. And then if you've shrunk down the information from your inputs into this embedding space, then you can now bring it back out again to the likely predictions. So this is a simple like one hidden layer architecture, but you can imagine if you had a neural network architecture with many different layers, you get these compressed representations all the way through the neural network where the architecture in a way is a little bit arbitrary. It's hard to say exactly which architecture of different amounts of layers or neurons is good. But in general, you do want to compress your data down into a space that's more easy to handle and learn on. So we know that language models like ChatGPT are extremely accurate. So here I asked what the iGEM UDEM team does, and it gave this output that was pretty good. And this is ChatGPT is a state-of-the-art language model that has been trained on pretty much the entire internet, all of the text downloaded from the whole internet. And from that, it's learned this probability distribution around almost any response or any question you can give it. It will give a nice high likelihood answer to your input text. Now, I wanted to ask everyone just what languages you know and see if there are people here who maybe know many different languages. So if you can, I think you can annotate the screen with by typing on it or something. Are they able to annotate or is the permissions?
I don't think so.
Oh, okay.
Perhaps in chat.
Maybe type in the chat. Okay, people are saying coding languages, C and C++, Python. How about just spoken human languages? English, Spanish, German, Czech, German, Spanish, French. So we have a mixture of many different languages and coding languages. I think that's a pretty good representation from a small group of people. So my point here was that no one said DNA, and DNA is a language that uh, all of life operates on. And um, if we treat it that way as a language, maybe we can actually learn it. So DNA is DNA, RNA, and proteins. They operate on these molecular languages that describe life. And the central dogma is the core tenet of modern molecular biology as to how all living cells store and process information in order to produce the functions that they use to survive. And it starts with DNA, which stores the information as a set of kind of instructions and parts. And then those instructions get transcribed into RNA, and then the RNA gets translated into proteins. And then proteins are the small molecular machines that do all of the functions that make the entire living world possible. So I'm going to start by focusing on DNA and genomes first. And this is a very simple four-letter alphabet, but despite only being four letters, it's has and encodes just all of the incredibly complex things that we still barely understand today. And a good fact that kind of helps grasp the enormity of of DNA and evolution. There are more bacterial viruses on Earth than there are stars in the universe. So scientists estimate there's around 10 to the 31 bacteriophages or bacterioviruses on the planet. And this is just one specific type of genetic entity. So you can imagine all of these genomes are floating around us, and there's just so much information to understand about them and different tools and technologies that we can harness from them. But it's very, very difficult. How do we navigate this extremely vast genomic space on the planet? And I want to see if people can guess here which sequence of DNA, number one or number two, is the real genome. Maybe type in the chat again. And it's okay if you get it wrong. I wouldn't be able to answer this question correctly. This is a very difficult question. Number one, number two, two, one. Okay, we're getting like a split of different guesses. Both. Yeah, I could be tricking you. It might be both. Okay, so it's clear that it's hard to discern. So actually, number one is the real genome. This is the gene part of the genome of rabies virus. The sequence on the right, it's completely randomly generated. I just coded a random DNA generator and it outputs nucleotides. Uh, but as humans, it's very hard for us to distinguish this difference. You can maybe tell some slight changes like biases in the real genome that are different than a random sequence. But in general, it's very, very unintuitive. And um, given that there are so many genomes on the planet that are just waiting to be explored, how could we possibly, as humans alone, navigate all of those genomes? And you can kind of think of this also as like this incredibly difficult and large cryptography problem where we're trying to decipher all of these genomes. This is a picture from The Imitation Game, which is a great movie based off of real events as to how Alan Turing deciphered codes during the World War. Um, and so DNA is a similar code that we should, as scientists, be trying to decipher because it's incredibly important for health, planet health, and technology. Up until now, we've pretty much mostly tried to establish these different hard-coded rules for genomes. So these are things that I was describing before, like the central dogma. You have reading frames of genes. We can describe different parts of genes and non-coding genes. But they can only take us so far because they don't always apply to every single genetic sequence. And um, there's just too much diversity for us to really apply these specified rules to every single unique DNA sequence. So what if we just try to learn it as opposed to us? Maybe is the genome a language that AI can learn? And this is coming back now to explaining how language models work. So yes, it is possible for language models to learn rules of DNA from millions of genomes. We have lots and lots of genomic data available to us, and now we can train these language models on them through the training process I was describing before. So EVO is a new type of language model. We're calling it a genomic language model, where it has been trained on millions of these genomic sequences. And the big advantage of training on DNA genomes is that you get all of this information encoded together that you wouldn't otherwise get if you trained on only RNA or only proteins or only coding sequences. Because DNA encodes all of these coding and non-coding elements like promoters, ribosome binding sites, genes, non-coding RNAs, etc. And so that's what EVO is. It was trained on millions of these DNA sequences. And really what it's doing here is learning, like I said earlier, a probability distribution around the language. So this is around the language of DNA. So you have inputs where you have some DNA sequence, and the model can predict the next nucleotide or the next letter in that DNA sequence. Um, and it's very important that it's doing this at the resolution of single nucleotides. Um, so, for example, if you pass in this sequence GC, TTAT, etc., it will predict the next base in that sequence, or it can also predict or just calculate likelihoods on an entire sequence that you pass through. So EVO was trained on pretty much all of the evolution that we've observed today. We have sequence databases of genomes from all of life, and this is just a visual of the sheer diversity of what EVO was trained on and all of these different phyla across each domain: bacteria, eukarya, and archaea. And this is extremely powerful. It works very well. So EVO can do things like predict disease mutations. This is data from the paper where um, they passed different genes with human disease variants through the model and wanted to be able to tell if EVO could score the likelihood of those variants being pathogenic or not. So these variants, we call them single nucleotide variants or SNVs. Some of them are pathogenic, like the one on the bottom here, or some of them are benign and they don't have any bad effect on the gene. And you can see that EVO scores non-single nucleotide variants highly, um, whereas it scores the ones encoding sequences a bit more lowly. So it's able to tell the difference between these, and is there's also data in the paper where it was applied to cancer mutations like BRCA1, and it works quite well too. So this is a very powerful function of the model. A question in the chat: is EVO like AlphaFold but for genes? AlphaFold is also a deep learning model. It's used to predict the structure of a given protein sequence. It's, in a sense, they're similar that they both use similar elements of neural networks as their architecture, but the task of AlphaFold is very different. AlphaFold operates on protein sequences. Um, AlphaFold 3 now can have different elements as well, but it's mainly proteins. And AlphaFold is trying to predict structures of proteins. Here, EVO is a generative model that can generate sequences of DNA or predict whether sequences of DNA are likely or not likely. Um, so the reason why that's a good function of EVO is because it's, from all of this training, it's essentially learned fitness, evolutionary fitness. So if EVO is able to score the likelihood of different sequences, you'd expect that high likelihood sequences are the ones that are very fit based on what it's learned from evolution. Whereas sequences that are not very fit or unlikely as a DNA sequence, it will score lowly or give low probability to those sequences. So this simple ability to understand fitness of pretty much any DNA sequence has very, very great implications for designing better technologies for synthetic biology. And I'll explain some examples as to how. So I guess synthetic biology is a very broad term. When I say synthetic biology, I'm referring to this intersection of engineering and biology where you are designing and building biological systems for useful things. So EVO can also design these multi-component genetic systems. It's able to generate long sequences. And if you prompt the model in the right way, you can specifically design tools that you're interested in. So EVO was able to design these entire CRISPR-Cas9 systems. CRISPR-Cas9, probably most of you know, is a very, very effective genetic engineering technology used to cut DNA and insert desired sequences. There are much more use cases for it, but that's how generally Cas9 works. And the reason why it's really, really powerful is because you can provide it a specific sequence that the protein complex will take and use to target any other DNA sequence that you're interested in. So Evo could design the different components of the CRISPR-Cas9 system, the protein coding sequence, as well as the non-coding RNAs that are used to create this targeting piece that allows it to target DNA sequences. So a bit more about how CRISPR works. The Cas9 protein has these two main domains, the H&H domain and the RUV-C domain. When paired with the non-coding RNA, the CRISPR RNA component is the part that you design your target sequence in, and then that will match the DNA sequence that you want to cut. So Cas9 will take the whole guide RNA to the target site in your DNA. And then with the H&H and RUV-C domains, it'll cut the DNA at that target site. And then after it's cut the target site, you can have other floating pieces of DNA around that you will specifically want to recombine into your target site through a process called non-homologous end joining, or sorry, homology-directed repair. And then non-homologous end joining is a natural repair pathway in cells where it kind of just repairs that cut site in the genome and it can introduce mutations. So CRISPR is very useful for specifically inducing mutations at sites of interest to study functions of different genes and things like that. In nature, CRISPR defends bacteria against their viruses. This is where we harnessed CRISPR-Cas9 from. So when a bacterial virus infects a bacterial cell, it'll inject its genome into the cell, and then the bacteria uses the CRISPR system to take pieces of that virus's genome and then use them as guides for the Cas9 complex to later cut the virus after it enters. So it's kind of like this innate immune defense system that bacteria have evolved and that we now use for very sophisticated genetic engineering. The Cas9 complexes that Evo designed turned out to be functional. So this is an experiment on the right where the Evo designed Cas9 was compared against the most commonly used SP-Cas9 that synthetic biologists use. And this was a basic cutting assay where the Cas9 was mixed with a target DNA sequence and then incubated over time. And after 3 hours, you can see at the top of the gel, there's some of the remaining target sequence, but it's been cut into two halves that have gone down in the gel. And you can see that on the very right, the Evo Cas9 also cut the target DNA sequence quite efficiently, potentially even more efficiently than the wild type SP-Cas9. And the bottom is just a schematic of what the structure of the Evo Cas9 looks like compared to the natural Cas9. So this is a very, very complicated design task that Evo was able to accomplish, and goes to show that training on a very large body of DNA sequences allows it to generalize and design synthetic biology tools like Cas9 and potentially even make them better. And if you scale this up, Evo can actually even design entire realistic genomes. So this is on the top, the M. genitalium bacteria genome, a visual of all of its different genes. And this is about a 500 to 600,000 nucleotide genome. Evo 1 and Evo 2, if they're prompted with a little bit of the beginning of the M. genitalium genome, you can let the model just start generating the next most likely DNA all the way up to 600,000 base pairs, which is a really, really long continuous sequence. And you can see that the model designs all of these functional or likely functional genes across this very, very long coherent DNA sequence. This, of course, wasn't tested in the lab. It's very hard to test a massive DNA sequence like this. We don't have the ability to really replace an entire bacterial genome yet. But when we have those experimental abilities someday, we can test whether or not AI can design long genomes like this in the lab. but at a computational level, they seem to be quite realistic. Um, so really anywhere that genetic design is possible for synthetic biologists, you can imagine that AI might be able to step in. This is just several examples of different synthetic biology applications for designing genetic circuits. You can use them for chemical synthesis pathways, or if you're imagining gene therapy using CRISPR, um, or if you want to design circuits that therapeutic bacteria might implement in the gut microbiome or something, or if you want to engineer smart plants, all of the genetic tools that are used in each of these different systems, you could use in a generative model like Evo to improve your designs or predict whether certain designs might be good or bad. So it helps narrow the massive DNA genomic space that I was referring to earlier down to a more tractable language that we can traverse around using AI. Now let's go to understanding proteins. I'm going to skip RNA today. There are RNA language models, but it's quite new. So protein language models are much more developed. And the proteome is a 20-letter, roughly 20-letter alphabet. There are some exceptions. So it's a much bigger alphabet than DNA. Um, and it, it's a very rich source of information for language models to learn from. It's a bit easier for them to learn from, and that's why protein language models are more developed than DNA language models today. A quick refresher on proteins. Hopefully this is review for everybody. The protein sequence, these different amino acids string together is what we call the primary structure, and they fold within the cell into substructures called secondary structures like beta sheets and alpha helices. And then those come together into a larger complex structure called the tertiary three-dimensional structure. And then quaternary structure is if you have multiple protein tertiary structures pieced together. And these are this is the foundation of all of the molecular machines that life uses, is these little chemical machines that form different complexes with different shapes and biochemistries that allow them to do everything in life. A goal for using protein sequences with machine learning models is to expand the possible protein design space. So you can think of it like this ocean of possible protein sequences, and evolution or native proteins have only sampled small islands or small parts of this big ocean. And then we have methods in the lab like directed evolution, where we can induce mutations in protein sequences towards a desired goal. And that has expanded the protein design space a little bit. But with protein design using machine learning and other computational models, we call this de novo protein design, where you can design entirely new to nature proteins that might have specific functions that are useful to us or to other benefits. And this is not to replace natural protein sequences. There are already an extremely large amount of functions encoded in natural proteins, but de novo protein design using computational models allows us to more rationally engineer what we want certain proteins to do and maybe do new functions that haven't existed in nature before. For example, this is a really cool idea where you could imagine designing enzymes or proteins that can degrade plastic. So if we have all of these landfills filled with tons and tons of plastic waste that are contaminating our environment, what if you could design proteins that can degrade this pretty quickly? So this was done in a paper published a few years ago. They used a machine learning model to recommend high probability amino acids in an enzyme called PETase, which is an enzyme that its native sequence can degrade plastic a little bit. They engineered it to degrade plastic much faster, and so on the bottom here, you can see that when they apply the engineered enzyme to a plastic tray, over the course of just a couple days, that plastic tray becomes completely degraded. This is a very, very good demonstration of what's possible if you use machine learning to recommend good functional mutations that we can then engineer in the protein. Another interesting use case for machine learning is to try to predict the evolution of viruses. This is very, very relevant, especially at pandemic time or for designing new vaccines. So if you can imagine the SARS-CoV-2 mechanism of entry, it uses this protein on its surface called the spike protein, which binds to the ACE2 receptor on our cells. And this is what is kind of the initial like lock and key mechanism that the virus uses to infect us. And it, at the kind of a more molecular visualization, this is happening between the receptor binding domain, or RBD, of the spike protein and the binding interface of ACE2. And so if we can break this down to a molecular language, it's possible we can actually train a model to predict likely or unlikely mutations of the virus and see if we can predict its next most likely evolution. And this is on the protein language space again. So this is a model called a variational autoencoder in the panel A, where you have lots and lots of amino acid sequences of the SARS-CoV-2 spike protein, and you train the variational autoencoder to compress the spike protein into a very, very small embedding space. So if you remember, I explained embeddings earlier. We're compressing information down into smaller dimensions. Here, we specifically compressed the information of these protein sequences down into 3 numerical coordinates. And then what we did was we plotted those 3 coordinates as a function of time, and then found the best fitting Gaussian process regression functions on those coordinates of the sequences, and then projected those functions into the future past the observed time of the training data. And then in the future, you get these new coordinates of each of these sequences, and you can use that as a proxy for the evolutionary trajectory of the viral protein sequence. And then you take those new coordinates and you pass them through the decoding part of the variational autoencoder, where it can then turn those numerical coordinates back into protein sequences. So this was a powerful concept using machine learning with a very simple neural network model called variational autoencoders. And the hypothesis here was that maybe by compressing viral protein sequences into a continuous numerical space, you can maybe map evolutionary trajectories on that space. And it worked pretty well. It was very close to predicting the most likely evolution of the virus. So we had projected the evolution up to 5 months into the future, and we got these 17 different likely variants that the model outputted. And you can see that sequence A, which was the most likely variant that the model predicted, was the most frequent virus in the training period. But then in the prediction period 5 months into the future, it went down, but its nearest neighbor, which was only 1 amino acid different from it, was the most likely variant. So the model was very, very close to predicting the evolution of SARS-CoV-2. And this is a preliminary work that can definitely be expanded upon. And hopefully someday in the future, when the next pandemic occurs, we can quickly use a model like this or other machine learning models to predict the evolution of the virus that we have a pandemic for, and then we can design our vaccines and treatments accordingly. The model also is generative in a way. It generated variants of the spike protein sequence that have never been seen in nature before. And so it would be interesting to study these sequences that the model thinks are likely, and be able to tell whether or not those are fit high fitness sequences. And maybe that can help inform the way we design our vaccines in the future too. These variants, if you use, so someone asked earlier about AlphaFold, you can use AlphaFold to model also the binding interactions between multiple proteins. So what we did was we used AlphaFold to predict whether or not these new variants of the spike protein bind the human ACE2 receptor. And they, based on this binding score called P.Q, they all bound the human ACE2 very well. So there's lots of potential here in a model like this, and I really hope that someday we can have better models doing things like this. So altogether, I hope now it's become clear that deep learning is helping us navigate the languages of life, whether it's DNA, RNA, or protein. And this very, very vast genomic space is more approachable, and we can pick and choose which parts of it we want to traverse and the ways we want to use it to create new technologies and new possibilities in synthetic biology. So that's my whole talk for today. I'm not sure how long I went, but I want to finish on this picture by Rene Magritte, which is a depiction of an artist painting a bird just by looking at an egg. And this is a nice representation of human creativity. And I hope that some of the ideas in today's talk you can use to apply to your creative ideas in synthetic biology or other research that you're pursuing. So I'm happy to take any questions about the presentation. And if I'm not sure how much time we have left, uh, but please feel free to ask anything, whether it's about the research or anything personal as well.